imagine, just for a second, that I hand you a manual. But, this isn't just any manual. It's this absolute behemoth of a book. And it contains 3.1 billion letters.
That is a lot of letters.
Right. And there are no spaces. There's no punctuation at all. It's written entirely in a foreign language that consists of only 4 letters. Oh. And then I look you right in the eye and say, hey, this book right here, it contains the exact step-by-step instructions for building a human being. Read it. I mean, where do you even start?
Honestly, you'd probably just want to throw it out the window.
Seriously?
Because it sounds like a literally impossible task. But that is the actual literal challenge a global team of scientists took on starting way back in 1990.
And that impossible task is what we are getting into today. Welcome to our latest deep dive. We are exploring one of the, frankly, most audacious scientific achievements in human history, the Human Genome Project.
It really is a marvel.
It is, and we are pulling from a really fascinating scientific reference report today. It's titled An Architectural and Operational Breakdown of Humanity's Genetic Blueprint. So our mission for you on this deep dive is to decode the highly technical process of how scientists actually read DNA.
Right.
We are going to simplify the incredibly clever chemistry behind it, and, we're going to reveal how researchers managed to locate the tiny fraction of functional genes hiding inside this vast landscape of non-coding genetic material.
Yeah, and it's a journey that really requires you to appreciate the era it happened in, I think.
Oh, for sure.
Because when you look at this 13-year international effort stretching from 1990 to 2003, coordinated by the U.S. Department of Energy, the NIH, and all these partners all over the globe, you realize pretty quickly that this wasn't just a biology experiment.
Right, it was way bigger than that.
Exactly. It was a massive computational and operational moonshot. I mean, they were trying to sequence 3 billion base pairs of microscopic DNA using technology from the early 90s.
Which is just wild to think about. I mean, consider the computers we were using back then. We're talking dial-up internet, floppy disks, those big beige towers with the turbo buttons on them.
Oh, yeah, the turbo buttons.
Right, and yet their primary directive was to identify all estimated 20 to 25,000 genes, sequence the entire 3 billion letter structure, and then dump all of that data into freely accessible public databases.
And that technological limitation of that era, that's the fundamental hurdle that shaped the entire project. Because before you can read a 3.1 billion letter manual, you have to figure out how to physically handle it.
Makes sense.
And sequencing machines back then, they simply could not process an entire chromosome sequentially from end to end.
Wait, really? Why not? Because I think the general assumption is, you just put the chromosome in a scanner. You start at letter 1, you just let it run to letter 50 million.
Yeah, I wish. But biology doesn't really play nice like that. A chromosome is an incredibly long, really delicate molecule. It tangles, it folds over on itself, and it is highly fragile. If you try to read a continuous string of millions of letters, the chemical reactions start to degrade, and the machines they lose track.
Oh, wow.
Yeah, the error rate goes completely through the roof after just a few hundred letters.
So they had to break it down before they could build it up.
Exactly. They deployed this strategy, and it's called hierarchical shotgun sequencing.
Shotgun sequencing. That sounds intense.
It is, kind of. The idea is basically systematic destruction. First, they took these massive human chromosomes and chopped them into large, manageable chunks. We're talking about 100 to 200 kilobases long.
Okay, so that's roughly, 100,000 to 200,000 genetic letters per piece?
Spot on, yeah.
Okay, let's unpack this for a second. It's like taking the entire encyclopedia of human biology, and rather than just sitting down and reading it, you're ripping it into distinct chapters.
That's a really good way to put it. But if you just rip up an encyclopedia, you basically just have a giant pile of paper, right? You have no idea what order the chapters go in.
It's true. That would be a nightmare.
So to solve this, they took those 200,000 letter chunks and cloned them into what are called bacterial artificial chromosomes, or BACs.
Wait, hold on. They put human DNA into bacteria.
They absolutely did, because bacteria are incredibly efficient biological copy machines. When you insert a chunk of DNA into a bacterial cell, every single time that bacteria divides, it faithfully copies that human DNA chunk.
Wow, okay.
So suddenly you have an unlimited supply of that specific chapter. But more importantly, they mapped these BACs onto the original intact chromosomes by identifying unique known landmarks.
And what are those called?
They're called sequence tag sites, or STSs.
Okay, so think of an STS as like a GPS pin dropped onto a specific page of our encyclopedia.
Yes, exactly. That's a great way to visualize it. So once they had all these massive chapters mapped out and pinned in the correct order, they took each individual BAC clone and physically sheared it.
Sheared it? Like with tiny scissors?
Well, they used sound waves, actually, or physical pressure, to shatter those chapters into tiny overlapping fragments. And these were only ranging from about 1,000 to 2,000 letters long.
Oh my gosh. So we've gone from the whole book down to the chapter, and now we're literally tearing those chapters into single sentences just so our magnifying glass can actually look at them.
Right, and those tiny sentence-sized fragments were then subcloned into even smaller delivery vehicles called plasmid vectors.
Okay.
And those were optimized for the raw chemical sequencing machines. This hierarchical approach, it solved the fragility problem, because you map the big pieces first, so when you shatter them into 1 million tiny pieces, you at least know the biological neighborhood they originated from.
Which brings us to the actual wall we have declined here.
Right.
We have these tiny sentence-sized fragments of 1,000 letters. But how do we physically see the chemical letter?
Great, that's the big question.
Because it's not like you can just put a strand of DNA under a standard microscope and just, read an A, C, T, or G. They don't look like alphabet blocks.
No, they don't. They just look like microscopic clumps of atoms. So to actually read the code, scientists had to trick the natural process of DNA replication.
Okay, how?
They used a method called automated Sanger-Dideoxy chain termination sequencing.
Okay, that is a very intimidating string of scientific words.
I know. Let's strip away the jargon. In your body right now, you have these biological machines called DNA polymerases, and they are constantly copying your DNA.
Okay, DNA polymerases, got it.
They read the original strand, and they build a matching copy by floating around and grabbing standard building blocks. These are molecules called DNTPs. And they just link them together one by one into a long chain.
So it's kind of like a blindfolded construction worker feeling around for bricks and just stacking them up into a wall.
Exactly. Now imagine we take that blind construction worker, the DNA polymerase, and we put it in a test tube with your unknown DNA fragment.
Okay.
We flood the tube with plenty of standard normal bricks. But we also mix in a small amount of special fluorescently labeled bricks.
Fluorescent bricks.
Yeah, these are the dideoxynucleotide triphosphates, or DDNTPs for short.
Okay, so our construction worker is reaching into a pile that has mostly normal bricks, but there are a few glowing, colorful bricks mixed in there too.
Yes, and those glowing bricks have a crucial, deliberate flaw. They lack an essential chemical piece called a 3' hydroxyl group.
Which means what, exactly?
Well, picture a standard Lego block. It has that little peg on top, right?
Right.
That peg is what allows the next block to snap onto it.
Oh, so if the peg is missing, you can snap that glowing brick onto the tower, but you can never snap another brick on top of it.
Exactly. The moment the blind construction worker accidentally grabs one of those fluorescent pegless bricks and adds it to the chain cannot grow any further.
Wow.
It acts as a chemical stop sign. The elongation process is instantly and permanently terminated.
Wait, so we are deliberately sabotaging the copying process?
We are.
That's wild. It's like stopping a train at every single track tie just to paint the final car a specific color.
We are absolutely sabotaging it. And because there are millions of copies being built simultaneously in this little test tube, and the incorporation of those stop sign bricks happens entirely by random chance,
Right.
you end up with this massive statistical soup. You create a population of synthesized fragments of basically every single possible length.
Let me make sure I'm picturing this right. Let's say my sequence is 100 letters long.
Okay.
In this test tube, one of the builders randomly grabs a glowing stop sign on the very first letter. So I have a fragment that is exactly 1 letter long.
Yep.
Another builder makes it 2 letters before grabbing a stop sign. Another makes it to position 85.
Spot on. You end up with a fragment of length 1, length 2, length 3, all the way to length 100. And the brilliant part is that each of the 4 DNA letters has a specific, unique color tag.
Okay, what are the colors?
Adenine is glowing green, thymine is red, cytosine is blue, and guanine is yellow, or sometimes represented as black.
So we have a soup of microscopic fragments, all different lengths, all ending in a specific color. But how do you actually sort them out? If they're all just floating in a tube together, you just see a muddy brown liquid.
Right, you do. So you force them into a race.
A race.
Yeah, using automated capillary electrophoresis. It's essentially this microscopic sorting tube filled with a dense gel matrix. You apply an electrical current to it, which pulls the DNA fragments through the gel.
Oh, I see.
And the friction of the gel means that the smaller the fragment, the faster it travels.
Like a physical obstacle course. Yeah. The one-letter train crosses the finish line first, then the two-letter train, then the three-letter track.
Exactly. And waiting right at the finish line is a laser.
Of course there's a laser.
Naturally. As each fragment exits the tube, separated perfectly by a single letter's length and size, the laser shines on it. The fluorescent tag glows, a sensor detects the specific color, and it creates a visual peak on a chart called a chromatogram.
Green, red, blue, green, yellow. And suddenly you are reading the genetic code, letter by letter, in perfect sequential order.
And what's fascinating here is the sheer computational mosaic required to put it all together afterward. That's what elevates this from just a neat chemistry trick to a true technological marvel.
Because you have so many pieces.
Millions of them. Phase 2 of this project generated millions of these short 500 to 900 base pair reads. So to assemble them, supercomputers had to run incredibly complex string matching algorithms.
Because we shattered everything at the very beginning, so now the computer has to find the overlapping edges to stitch them back together.
Right.
So if one sequence ends in ATCGJ and another sequence starts with CGGGA, the computer realizes they overlap at CGGA and just glues them together into a larger block.
Yeah. Exactly. And they call those larger contiguous sequence blocks contigs.
Contigs, got it.
The supercomputers bridged all the gaps, building these extensive sequence scaffolds across those initial BAC frameworks we talked about earlier. And by 2003, this algorithmic stitching achieved a staggering 99.99% accuracy.
That is incredible.
Delivering a reference genome that was 99% complete for the gene-rich regions of our DNA.
That is just mind-blowing logistics. But here's the thing that gets me. Having a 3.1 billion letter string assembled in a computer is really only half the battle.
Oh, absolutely.
Because if I hand you a 3 billion letter text document with no spaces, no bold text, and no chapter headings, finding the actual instructions is a total nightmare. It's just a wall of noise.
Which is why having the sequence was really merely the prologue. The next massive challenge was phase 4, which is annotation. Right. Annotation is the systematic process of scanning this vast chemical landscape to pinpoint the specific biological coordinates. You have the raw map, but now you need to draw in the cities, the highways, and the stoplights.
So how do you actually find the usable instructions like the genes hidden inside that massive wall of text?
Well, supercomputers scan the DNA looking for highly specific chemical patterns. Primarily, they search for protein-coding exons.
What are those?
These are the sequences that directly translate into the amino acid strings that build every physical protein in your body. The algorithms look for what are called open reading frames, or ORFs, which are stretches of DNA bracketed by start and stop codons.
Oh, like finding a capital letter at the beginning of a sentence and a period at the end? That tells you, hey, there's a functional instruction here.
That's a perfect analogy, but the most shocking revelation of the entire annotation phase wasn't how they found the genes, but really how few there were.
Wait, what do you mean?
When they mapped all these protein-coding regions, they discovered that these functional protein-making genes comprise less than 2% of the entire human sequence.
Less than 2%?
Less than 2%.
Wait, we'd spent 13 years, an international coalition of scientists, and billions of dollars sequencing 3 billion letters, and only 2% of it actually makes the proteins that build us.
Yeah, it was a paradigm-shattering moment in biology. For decades, many scientists had looked at the massive amounts of non-coding space and just assumed it was evolutionary junk mail. They literally called it junk DNA.
I remember that term, junk DNA. So I do the math here. Well, if less than 2% is actual genes, we are missing over 98% of the genome.
Yep.
What on earth is taking up all that remaining space? Is it really just useless filler?
Calling it junk was honestly one of the biggest misconceptions we've ever had in science.
Really?
Think about it. A complex system needs way more than just raw building blocks. It needs a highly sophisticated operating system. That 98% is the control board.
Okay.
According to the report's structural composition profile, about 24% consists of introns and regulatory blocks. And these are non-coding sequences embedded right inside the genes.
So they are inside the gene, but they don't actually make protein.
Correct. They control the structural splicing and stabilization of the gene itself. Then you have promoters and enhancers. Think of them as biological interfaces.
Like switches.
Exactly. They interact with cellular machinery to dictate exactly when a gene should be turned on, in what specific tissue, and for how long. I mean, you have the gene for making stomach acid in every single cell of your body, including your eye.
Oh, yikes.
But you really, really only want that gene turned on in your stomach. The promoters and enhancers ensure that happens.
Okay, that makes total sense. So the 2% are the light bulbs, but the promoters and enhancers are the wiring and the light switches.
Precisely.
But that still only accounts for around 26% of the genome. We still have a lot of missing space.
Well, let's keep going. About 24.5% is unique non-coding DNA. And a massive 50%, fully half of your genetic code, consists of repetitive DNA elements.
Half of my DNA is just on repeat.
Yes. This includes interspersed repeats, highly compressed structures called heterochromatin, and transposable elements, which are sequences that can actually move around the genome.
Whoa, back up. Transposable elements that move around.
Yeah.
Are you saying parts of our DNA literally detach and travel to other parts of the sequence?
That's exactly what they do.
How does that not completely destroy the code?
I mean, it sounds chaotic, doesn't it? But over millions of years of evolution, these moving elements have become a dynamic scaffolding. They actually drive genetic variation and evolution.
That's wild.
And that highly compressed heterochromatin, it dictates the physical, structural integrity of the chromosomes themselves. If you don't have that dense packing material, the chromosome literally falls apart during cell division.
Wow, that completely flips the perspective. It's like looking at a bustling city. The buildings, the actual protein coding genes, they only make up a small percentage of the physical space.
Right.
But the rest is the roads, the power lines, the traffic lights, the structural foundation of the earth underneath it all. If you take away the 98% infrastructure, the 2% of buildings just can't function.
You've captured it perfectly. It is a highly regulated control environment. And mapping both the buildings and that infrastructure fundamentally rewired the economics and the entire future of global medicine.
I really want to dig into the math on the economics, actually, because this is where the timeline just blew my mind.
Oh, the economics are staggering.
The report highlights this equation for sequencing efficiency. Cost per base pair equals the net operational overhead divided by the total nucleotide throughput.
Basically, how much does it cost to run the laboratory divided by how many letters you can read simultaneously?
And the trajectory of that single equation represents the true legacy of the project, really. Because in 1990, the cost hovered near $1 per base pair. You have 3.1 billion base pairs. That is $3 billion just to read the sequence once.
Yeah, an astronomical sum.
But today, because of next-generation sequencing platforms, that metric has plummeted to fractions of a microcent. You can get a full human genome sequence today for under $100.
It's unbelievable.
We took a multi-billion-dollar global moonshot and reduced it to the price of a decent pair of sneakers. If cars dropped in price that much, a Ferrari would cost a nickel.
The scale of that cost collapse is genuinely unprecedented in scientific history. And it is what shifted medicine from a reactive discipline where doctors wait for you to present symptoms and then kind of guess at the treatment to an anticipatory molecular scale paradigm. It gave rise to pharmacogenomics.
So we're no longer just guessing which drug works. We're tailoring the treatment to your specific genetic code.
That is the ultimate promise of precision oncology and modern diagnostics. By cross-referencing a patient's individual profile against the baseline reference sequence generated by the Human Genome Project, researchers can isolate the exact genetic variants responsible for rare pathologies.
That is so cool.
They can target a cancer tumor at the molecular level based on its specific mutations.
But none of that innovation, none of those startups or medical breakthroughs would be possible if the initial data had been locked away behind a corporate paywall.
No, definitely not.
Which brings up something I found deeply inspiring in the source material. The Bermuda Principles of 1996.
Yes. The Bermuda Principles cannot be overstated in their importance. In 1996, the scientific leaders of the project came together and formulated a strict mandate.
What was that?
All raw genomic data generated by the project must be made publicly available on the internet within 24 hours of generation.
Within 24 hours. No hoarding data for academic glory. No patenting the basic building blocks of human life for corporate profit. They just threw the doors open to the entire world.
They recognize that the human genome belongs to humanity. That uncompromised open-access database became the permanent structural framework for everything that followed. The functional gene editing vectors like CRISPR, the advanced molecular diagnostic tests deployed worldwide today, they all stand on the shoulders of that open data.
So let's look at the big picture of this incredible journey. We started with a 3.1 billion letter manual that was impossible to read end to end. We had to physically shatter it into tiny fragments, clone it into bacteria, and shear it into microscopic pieces.
Yep.
We used fluorescent pegless stop signs to sabotage the copying process so we could sort and read the letters by color in a microscopic race. Then we used supercomputers to stitch those colorful overlapping fragments back together.
It really sounds like science fiction when you summarize it like that.
It does. And when we finally read the wall of text, we discovered that the secret to human life is actually a tiny 2% needle of functional code, hidden inside a vast, intricate 98% regulatory haystack.
It is a stunning realization of our own biology. And, it has direct implications for you, the listener.
Absolutely.
The next time you or a loved one gets a molecular diagnostic test or a targeted medical treatment, you are directly benefiting from this open source blueprint. It is quite literally the foundation of modern human health.
It really is. But before we wrap up, there is one final detail from the timeline that I want to leave you with to mull over.
Oh, this is a good one.
Remember how we said the original project had to chop the DNA into tiny 1 to 2 kilobase fragments to read it?
Right.
Well, because of that shattering process and the algorithmic limitations of trying to stitch together those massive 50% repetitive sections we talked about, the famous 2003 genome wasn't actually 100% complete. It had an 8% structural gap.
A gap that remained a mystery for nearly 2 decades.
Yes. It was only in 2022, with the telomere-to-telomere consortium using new long-read technologies, that the final 8% was filled in.
Exactly.
So if our new technology is finally allowing us to read massive lengths of DNA without having to violently break it apart first, what undiscovered genetic secrets might be hiding right now in the intricate repetitive structural spaces we previously couldn't put back together?
It is a thrilling frontier. We may finally have the complete manual, but we're just barely beginning to learn how to read between the lines.
There is always another layer to uncover. Thanks for joining us on this deep dive. Keep asking questions, keep wondering, and keep digging deep.